All right, class, so welcome to Monday, January 31st. Hope you guys had a great weekend. Here we are. We're going to finish chapter 3 today in lecture. So we'll finish chapter 3 and we'll probably start chapter 4, which is on tissues and their function. And then this lecture exam, which we're supposed to have on Wednesday, I'm probably going to push off to Monday. So you guys have a little more time to study these chapters. So what we'll do, we'll finish chapter 3 today, we'll start chapter 4, and then we'll finish chapter 4 on Wednesday. As well, on Wednesday, you can come with any questions. Maybe I'll look through the exam and give you a couple of pointers or key things to study for over the weekend. But that Monday then, which will be February 7th, that will be our first lecture exam. So I'm just moving the lecture exam on chapters 1 to 4 scheduled for this Wednesday. I'm just going to move it to Monday to give us a chance to this week to finish up chapters 3 and 4. And I think that's all I wanted to mention. So today in lab, if you have lab on Monday, you'll notice that we're just getting through 2 exercises. So you have no lab activities to do today in lab. Thursday or Wednesday lab, people from last week know that. And then Wednesday lab, you guys will start kind of a new section then with their lab activity with the integumentary system. Okay, so I just wanted to mention that. And again, if you joined us late, this lecture exam scheduled for Wednesday, I'm going to push to Monday to give us this week to finish chapters 3 and 4. So this Monday, this exam will be a week from today. And again, for that lecture exam, we'll go over how lecture exams work on Wednesday. But this exam, you have to show up to Zoom. You need to come with your video turned on so I can see your full face completely lit up before I give you that exam link, but it's all through Google as well. I'm sorry, let me try to, getting a lot of notifications here, so I'm going to try to turn that off. All right, so let's jump into where we kind of left off. So we're in chapter 3, we're learning about cell structure and function. So hopefully you guys can see my lysosome slide up there. This is where we're at in terms of this. And then we'll also start chapter 4 on tissues. Okay, so here we are in lysosomes. So if you remember, we just talked about the Golgi apparatus, which creates different vesicles that will then be sent to various parts of the cells. And some things that the Golgi apparatus creates are lysosomes. Or vessels for lysosomes to digest. So the Golgi apparatus might form some kind of garbage bags that get sent to the lysosomes. And the lysosomes will contain a variety of enzymes that function in intracellular digestion. So lysosomes will also be formed from the Golgi, but the big thing about lysosomes is they help with intracellular digestive processes. Vesicles formed by endocytosis that are being brought into the cell could fuse with the lysosome, and then the lysosome would then kind of secrete its enzymes in it to break it down. And one example of this is when a white blood cell phagocytizes or eats bacteria up. So as a way it kind of eats it up to get rid of it. So that's an example of a lysosome. So here's a look at lysosome action. You can see how a vesicle will form kind of within the wall. It will form this like vesicle or sac with whatever's inside of it. And then the lysosome will fuse with it. You can see how the lysosome comes from your Golgi because your Golgi helps to create lysosomes as well. The lysosome has special digestive enzymes inside of it that it'll secrete into this kind of vesicle. And then, the digestive enzymes that it does secrete will break down and digest anything that was taken in by the cell. So that's the action of lysosomes. They're kind of like your cell's garbage collection system and digestion. Peroxisomes are similar to lysosomes, but they're very specific in what they break down. So specifically peroxisomes, they also contain enzymes. They're like little vesicles, but specifically they break down fatty acids, amino acids, and hydrogen peroxide. So they're a little like stronger. Hydrogen peroxide will always be a byproduct of fatty acid and amino acid breakdown, and it can be toxic to the cell. So it's really important that peroxisomes get rid of that. And the specific enzymes in peroxisomes break down hydrogen, which again, as a part of hydrogen peroxide can be toxic. So your peroxisomes help get rid of that toxicity. Mitochondria are a big organelle because these are your cell's powerhouses. So mitochondria, singular, it's called a mitochondrion. They're small organelles responsible for producing considerable amounts of ATP by aerobic, which means with oxygen metabolism. That means in the presence of oxygen, mitochondria can create a lot of ATP or energy using glucose as its kind of starting point. And we get glucose from all the food we eat. Mitochondria have two membranes. So there's like an inner membrane and an outer membrane, and they're separated by a space. The outer membrane is nice and smooth, but the inner membrane has numerous folds to it called cristae, which will project into the interior of the mitochondria. The material within the inner membrane is the mitochondrial matrix and contains enzymes and its own DNA. And cells with a large energy requirement have more mitochondria than cells that require less energy. So for example, your muscle cells will have a lot of mitochondria because they need energy constantly to contract. And you'll have some cells in the brain that require lots of energy as well. So here's a look at what a mitochondria looks like. You can see this double membrane. The outer is nice and smooth, but then the inner membrane makes these folds. And those folds are called cristae. They're lined with different enzymes, but that's the inner membrane. It makes these folds called cristae. And the matrix refers to the deepest inside those folds, kind of the innermost part of the mitochondria is the matrix. And when we go through cellular metabolism, which is a really complex, fun process about how your cells turn glucose into energy, we'll look a lot into the mitochondria because that's where this occurs. So underneath the microscope, again, TEM, this is a transmission electron microscope. So the only way you can get a picture of this zoomed in is if you kind of pump electrons through the image. And this is looking at a cell or something 34,000 times magnified, which is pretty amazing. We can see something that small, but this is a look at a mitochondria, and you can see the folds from the cristae within that mitochondria. Then the cytoskeleton. So just like we have a skeleton of bones that gives us internal framework, your cell has a cytoskeleton as well. Again, the prefix cyto refers to cell. It consists of different proteins that support the cell, hold all the organelles in place, and then enable the cell to change shape and helps it divide as well. These protein structures, there's kind of three main groups. We have microtubules, microfilaments, and intermediate filaments. Those are kind of the three main groups of your cytoskeleton. Microtubules will be hollow, formed from protein subunits. The microtubules perform a variety of roles, including helping to support the cytoplasm of cells, assisting in cell division, and forming essential components of certain organelles like cilia and flagella. And cilia is an extension of the plasma membrane, and flagella helps with movement of the cell. And usually there's just a single flagella per cell. And the only human cell that has a flagella is a sperm cell. So those are made of microtubules. Microfilaments are much smaller. They're smaller fibrils, also formed from proteins. They just support the cytoplasm. They give the cell its shape. Some are involved with cell movement, and specifically microfilaments in muscle cells help muscle cells to shorten or contract. And we'll go through that when we get to the muscular system, how your muscles contract using these microfilaments. Intermediate filaments are fibrils formed from proteins. Again, they're smaller in diameter than microtubules, but larger in diameter than microfilaments. So the intermediate filaments are kind of the in-between size. They also provide mechanical support to the cell, and a specific type of intermediate filament is found in keratin, which we'll find out in the next chapter is an important protein associated with your skin cells. So those are intermediate filaments. So here's a look at the cytoskeleton. And again, the cytoskeleton, you can kind of see the microtubules have the largest diameter. Intermediate filaments are in between, and then the microfilaments are kind of the thinnest in the diameter. And, all kind of work to give the cell structure and shape. Each one has kind of a different purpose, but in general, they all work to give the cell structure, shape, help with movement, cell division, contraction if it's in a muscle cell. So those are all the cytoskeleton elements. You can see how they form kind of like a bridging network. And again, this is a scanning electron microscope under 60,000 times magnified. A microscope look at a cell showing those cytoskeleton elements, which again, I think it's always fun to see an actual picture and not just an artist's view of these things. Okay, centrioles are very important. The centrosome is a specialized area in the cytoplasm close to the nucleus where microtubule formation occurs. So we're kind of in the centrosome area. It's just a special area within the cell. It will contain two centrioles. So centrioles always come in pairs, and they're normally oriented perpendicular to each other. Each centriole is a small cylindrical organelle composed of microtubules, and the centrioles are involved in the process of mitosis. So the centrioles, they're in pairs. They will separate and go to opposite sides of the cell. And then from there, the mitotic spindle forms, which are like the fingers that reach up and pull the chromosomes apart. So those are your centrioles. And they're made of microtubules, which are those protein filaments that we just talked about. So here's we look at one centriole, and you can kind of see it's made of triplets, kind of are surrounding kind of a star-like structure. And again, this is just one, so you would have another one kind of perpendicular to it. Again, underneath the microscope, you can see a centriole and what it looks like. Cilia then is a projection from the surface of certain cells. Specifically, we'll see cilia projecting from cells lining in your respiratory tract because they help to move materials across the top of the cells like mucus. Cilia are cylindrical structures. They extend from the cell. They're composed of microtubules, and we'll see cilia again in the next chapter when we talk about connective tissue or epithelial tissue. And then flagella have a structure similar to that of cilia, but they're much longer. You usually have one flagella per cell. And since we're talking about human anatomy and physiology, in humans, sperm cells are the only cell that has the flagella. They have one of them, and they help propel the sperm cell for movement. Microvilli is another specialized extension of the cell. They're supported by microfilaments. They are smaller than microvilli, and they do not actively move. They're numerous on cells that have them, and they help to increase the surface area. So it kind of think of like a hairbrush. The microvilli, when they're on the surface of their cells, is often called a brush border because there's so many of them, and it helps to just increase surface area on specific cells that line your intestines, your kidney, and other areas of the body where absorption is very important. So in your intestines, the microvilli help to absorb digested foodstuffs. In your kidneys, microvilli will help to absorb or reabsorb things back into the bloodstream. All right, and then whole cell activity. A generalized cell, and its characteristics will always be determined by the type of proteins that produce them. So the proteins produced are in turn determined by the genetic information in the cell. And information in the DNA provides the cell with a code to make those proteins for a specific cellular process. So the genetic code, or your DNA, provides kind of the blueprint or instructions for creating the proteins that will then tell a cell what it's going to do, because we have so many different types of cells in the body. You'll tell a skin cell what to do based on what type of DNA creates the skin cell protein. Cells in your digestive tract will have different DNA to create proteins that determine that cell. So we need to understand DNA and a little bit of this process of what we call protein synthesis, how we turn DNA into protein that tells that cell what it's going to be. So we'll talk about DNA, and these next couple slides will go through protein synthesis. During our break before lab, I'm going to post a really good video that will kind of be an animation of protein synthesis that I really want you to watch when we're done with lecture, because that'll kind of put it into an animated version of what I'm about to explain and talk about. So DNA, again, contains the information or the blueprint or the set of instructions that directs protein synthesis. And that is a process called gene expression. When we turn the genes or the information in DNA into a protein. And again, the protein again will determine what that cell does. A DNA molecule consists of nucleotides joined together to form two nucleotide strands that are connected and resemble a ladder twisted around an axis. And each nucleotide consists of a 5-carbon sugar, a phosphate group, and a nitrogenous base. Each nucleotide on kind of one side of the DNA strand has a specific bonding pattern to another nucleotide on the opposite strand, so that only kind of the base pairs that are complementary will join together to join these two strands of DNA together. And a gene will just be a specific sequence of nucleotides that provides a chemical set of instructions or a blueprint or a genetic code for making a specific protein. And this process is called gene expression, or also known as protein synthesis, and it involves two really simple steps: transcription and translation. Transcription is the process where we turn or copy our DNA set of instructions into messenger RNA. So a type of RNA that's like the messenger that'll carry those set of instructions out of the nucleus. Translation then involves the messenger RNA that is carrying those DNA instructions, and it uses that messenger RNA then to produce a protein. And where do you think this occurs? Outside the nucleus, there was an organelle that creates proteins. It starts with an R, I, B. Your protein synthesis occurs in ribosomes. So this kind of messenger RNA will then take that set of instructions that it copied from the DNA, and it gets fed into one of these ribosomes to then produce a protein. And it's really simple. Those are the two main steps for gene expression or creating proteins, protein synthesis. Then we'll go into each of those two steps into a little bit more detail. So transcription takes place inside the nucleus of the cell, and DNA determines the structure of mRNA through transcription, because remember, mRNA is just a copy of the instructions that are found in DNA. During transcription, specifically, the double strands of the DNA segment will separate. DNA nucleotides of the gene will pair with RNA nucleotides that form the messenger RNA. And I'll show you a picture of this in 2 more slides. These are the bases in DNA. So these bases kind of, again, they bond together to keep the DNA the strands nicely put together. Thymine, adenine, cytosine, and guanine. And guanine always pairs with cytosine, those base pairs, and adenine always pairs or is attached to a thymine base pair. Messenger RNA, or mRNA, is a little bit different in that it contains adenine, cytosine, or guanine, but instead of having a thymine base, it's replaced with a uracil base. And again, the uracil will always pair with adenine, and the cytosine will always pair with guanine. So in here, this describes the pairing with specific RNA nucleotides. So for example, thymine in DNA always pairs with adenine. DNA's adenine would always pair with RNA's uracil. Cytosine pairs with guanine, and guanine pairs with cytosine. Remember, RNA is different because instead of having a thymine base pair, it gets replaced with a uracil base pair. So normally, thymine would pair with adenine always in DNA, but when we take a adenine base pair from DNA and pair it with an RNA base pair, it becomes a uracil. So let's take a picture and look at this. So this is again transcription. So this is the first step in creating a protein. And again, this first step is where we take the instructions in DNA. I'm terrible at drawing on my mouse pad here. It's where we take the set of instructions found in DNA and we make a copy of them, and we make a new kind of strand or a set of instructions, and that's called a messenger RNA molecule. So down here, you can see the DNA, this kind of helical wound pattern of 2 kind of ladders pulled together. And you can see here that, again, the 2 strands of DNA are held together by these base pairs. And you can see here that cytosine yellow will always pair with green. So that's cytosine and guanine. Thymine will always pair with adenine. That's red and blue. That's found in DNA. And then the uracil, the last base pairing that you see, the uracil binding with adenine, that's found in RNA. Remember, when RNA doesn't have thymine, it changes it into uracil. So these kind of base pairs or nucleotide pairing, those are important. They're complementary, so only certain base pairs bond or bind with each other. And again, this base pairing is important because it keeps the two strands nicely pulled together. They're kind of pulled together with hydrogen bonds, which are very weak. So in an example like transcription, these bonds can be broken really easily. So here we see the segment of DNA has been opened, and we can see in gray here, this is the new messenger RNA strand that is forming. And what happens is when the DNA strand opens, another strand of messenger RNA forms, and you can see how the complementary base pairs are starting. Uracil with adenine, cytosine with guanine. You can see an adenine with a thymine, but again, the thymine is on the DNA strand, so that's why it's thymine. And the uracils will always be on the RNA strand because that's a uracil is a base pair only in RNA. So this messenger RNA strand that is forming here, it's basically just, it's making a copy of that DNA strand, but that messenger RNA strand is going to take that copy or set of instructions to create a protein, and it'll take it outside of the nucleus. So that's transcription, making of messenger RNA, or just making a copy of the DNA set of instructions to take out to a ribosome to create a protein. All right, translation then is the second step in protein synthesis, and this occurs in the cytoplasm. So transcription, that process of reading the DNA, creating the mRNA strand, occurs in the nucleus. And then translation occurs in the cell cytoplasm when the messenger RNA with its set of instructions copied from DNA will exit the nucleus through one of the nuclear pores. And then this is the big thing, the messenger RNA attaches to the ribosome, because remember the ribosome is our big organelle in the cell that creates protein. The messenger RNA kind of feeds itself through a ribosome, and then the ribosome reads the strand and starts to create protein from that. How does the ribosome read the strand of messenger RNA? Well, it takes a codon, which is 3 nucleotide bases at a time on the messenger RNA, and they're read by what we call an anticodon on another molecule called transfer RNA. So transfer RNA kind of comes to the ribosome that's reading this messenger RNA strand, and transfer RNA is called transfer because then it'll go and get an amino acid that matches that codon, and it reads it with its anticodon. So transfer RNA, tRNA, will transport, that's why it's called transfer. It will go out and get a specific amino acid from the cytoplasm, and it'll take it to the ribosome and messenger RNA complex to start to initiate the form of a polypeptide chain. Remember that proteins are just chains of amino acids. So polypeptide, peptide means amino acid, poly means many. So this transfer RNA molecule is bringing all these amino acids and starting to link them together to form a chain, a protein chain. And the process continues until the entire polypeptide is completely formed. So here we have translation of mRNA in protein synthesis. So again, the mRNA is carrying the set of instructions from the DNA. So it exited the nucleus. The messenger RNA strand gets fed through this ribosome subunit, or this ribosome. So the purple is showing your ribosome. Remember, it's made of 2 subunits kind of put together. The messenger RNA strand gets fed through, and the codon is 3 nucleotide base pairs at a time. It's read by what we call an anticodon, which is a part of the transfer RNA molecule. So here's the transfer RNA molecule bringing an amino acid, that's the little green ball, and the little amino acid matches that codon that's being read on the mRNA strand. And each codon is specific for an amino acid that to bring in. And we figured this all out so we can actually go in and know the base pairing for each codon and the codon for each amino acid. So in things like a mutation or a genetic disorder, if one base pair is off, instead of bringing the right amino acid, the transfer RNA will bring the wrong one, and all of a sudden you've created a wrong protein, which could lead to disease or disorder. That's with mutations. If this stuff really interests you, I would encourage you to take genetics and continue on, because genetic coding, all about this genetics and base pairing is kind of like the hot topic in medicine right now, and how to kind of be able to predict disease based on your genetic code. But let's get back to this first of all, just trying to keep you excited and interested. So here we have the transfer RNA bringing an amino acid, and what it's done is you can see that the amino acid is here, and another transfer RNA molecule is bringing another amino acid, this one in blue. And again, we have 20 different types of amino acids, and we know all their names. And we've seen how we're starting to form this chain. So we see how the first transfer RNA molecule, after it brought its green amino acid, plops it on top of the blue one, and then it'll be released to go out to maybe get another amino acid or read the next codon. So because of this, we start to form this chain of amino acids. And when the chain of amino acids is complete, that forms the protein. And then that whole entire chain would be released from the ribosome, and that protein would go on and do what it's supposed to in that cell. So that's translation, the idea of bringing a messenger RNA strand, which is a copy of the directions from DNA, to create a protein, and in the ribosome, it's being read by transfer RNA molecules that bring in specific amino acids to match the codon. Simple version. I'm also going to again post a video that will give you an animated version. So here's just a good, again, really simplified version of an overview of gene expression. So we have DNA containing the information to create proteins. The messenger RNA strand is a copy of that DNA that is made, and the messenger RNA strand will then leave the nucleus, enter into the ribosome, so it kind of gets fed through. And then these codons, these 3 base pairs at a time, are read by a transfer RNA molecule that brings a specific amino acid. And these amino acids are joined together to form a polypeptide chain. When it's complete, that protein will be done, and that protein can then go on and do its function in the cell. All right, and then the cell cycle. So the cell cycle describes the growth, development, and cellular division. So just like humans go through kind of a lifespan, the cell goes through a cycle versus how it kind of functions. So during growth and development, cell division occurs to increase the number of cells or to replace damaged or dying ones. And this cell division involves a cell cycle. And we kind of divide your cell cycle into 2 major phases. The non-dividing phase, which is most of the time what your cells are in, it's called interphase. It's just your cells carrying out normal everyday functions, depending on which cell they're located in the body. And a cell dividing phase termed mitosis. So mitosis is specifically when one cell will divide and become 2, but the majority of a cell's life or cycle will be spent just in normal, everyday activities called interphase. So a cell spends most of its life in interphase performing normal functions. During interphase, DNA will be replicated, or DNA will be made. You'll make a copy of DNA so that when the cell divides, one of the original DNA can go to one daughter cell and the copy can go to a new daughter cell. So you'll make genetically identical daughter cells from one single cell. The 2 strands of DNA during cell division will separate from each other, and each strand then serves as a template for the production of a new strand of DNA. So we get into kind of DNA, again, separating, but in the purpose, again, of creating two new strands. Nucleotides in the DNA of each template strand pair with nucleotides that are subsequently joined by enzymes to form a new strand of DNA, and the sequence of nucleotides in the DNA template determines the sequence of nucleotides in the new strand of DNA. What this means is just that you're forming, you have one kind of your DNA of the original cell, you're separating it so that each new kind of DNA of the daughter cells will contain like an original strand and one new strand. And that is just to ensure that you have two genetically identical pieces of DNA being passed to the new cells. And that's important because if you're making new skin cells, you want them to be genetically the same so that each skin cell will have the set of instructions to continue doing what it's supposed to. Replication of DNA gives two identical chromatids joined at a centromere, and that forms one chromosome. So if we think of a chromosome kind of like an X-shaped structure, where you have these two kind of strands, and a chromosome is just kind of a really tightly wound DNA. This is one chromosome, but it's made up of these two strands that come together, and they come together at the middle. It's called a centromere. So here's DNA replication, and kind of similar to transcription, your DNA will separate where the old strand will provide kind of the template for a new strand to come up and form. So we're going from, again, one DNA, the original DNA molecule, to two new DNA molecules. And again, these two new DNA molecules will be exactly the same as the original because they're using the old strand as the template on both sides. And all the new strand needs to come in is bring the right base pairs, again, the right base pairs that are complementary with each other. You'll see them all joining, cytosines with guanines. So you'll see red and greens joining together, and then thymines with adenines. You'll see blue and reds coming together. So again, you're making two identical DNA molecules from the original one. And that's to prepare for cell division. So this process of replicating the DNA, this will occur in a normal interphase of the cell cycle, but it will occur in getting that cell ready to divide so that each new daughter cell will be genetically identical to the parent. Each human cell, except sperm and egg, we'll talk about those in a little bit. Each human cell contains 23 pairs of chromosomes for a total of 46, and the sperm and egg contain half that amount. So all of your human cells, they're called somatic cells, contain 46 chromosomes. However, sperm and egg contain half that amount, 23. And that's because the sperm and egg will have kind of the goal or, the potential to join and form one unit that would have 23 plus 23 is the total 46 chromosomes. But the sperm and egg are the only cells in the body that contain half the number of chromosomes. Otherwise, all your other cells in your body contain the full amount. One pair of chromosomes are what we call the sex chromosomes, which consist of two X chromosomes if the person is a female, or an X and a Y if the person is a male. So that determines your genetic gender if you have two X's or an X and a Y is a male. So mitosis. Mitosis is the process of forming two daughter cells from one parent. And we divide mitosis into 4 phases: prophase, metaphase, anaphase, and telophase. And if you remember PMAT, you'll remember those phases in order. And these 4 phases, this is sometimes a good matching test question where you would see the phase, prophase, metaphase, anaphase, telophase, and then on the side, descriptions of each phase, and you would just have to match what happens in each phase with the name. So just keep that in mind. You'll have to understand kind of what happens in each phase here. So prophase, during prophase, the chromatin condenses to form chromosomes, and chromatin is just your loosely coiled DNA. Chromosomes will be very tightly wound DNA, so it forms those nice X-like structures of a chromosome. Microtubules, termed spindle fibers, form and assist to breaking the centromere between the chromatids that form the chromosome to move the chromosomes to opposite sides of the cell, and the nuclear membrane dissolves. So during prophase, again, this is the first step. What we're seeing during prophase happen is we're seeing chromosomes form as they're getting ready to be pulled apart, because that makes it easier to separate out the DNA. And we won't have a nuclear membrane anymore. So normally you see kind of a membrane or a boundary around the nucleus. During prophase, that will dissolve or go away. Metaphase is the second step, and metaphase is a really easy one to remember and see, and I'll show you pictures of this in a couple of slides. During metaphase, chromosomes align or line up really nicely near the center of the cell, and the movement of the chromosomes is regulated by the attached spindle fibers. So these chromosomes, which are all X shapes, will line up in the center of the cell because then they'll be pulled apart. But metaphase is just the lining up of the chromosomes in the center of the cell. Anaphase, the chromatids will separate, and each chromatid is called a chromosome, and each of the two sets of 46 chromosomes is moved by spindle fibers toward the centriole at opposite poles of the cell. And at the end of anaphase, each set of chromosomes has reached an opposite pole of the cell, and the cytoplasm begins to divide. So we'll pull apart these chromosomes, and the cytoplasm will start to divide during anaphase. And again, I'll show you a picture of this, which will help kind of visualize this process a little bit more. Telophase, the chromosomes in each of the daughter cells become reorganized to form two separate nuclei, a nucleus in one and a nucleus in another, one in each newly formed daughter cell. The chromosomes will begin to unravel again and resemble their normal phase during interphase. And following telophase, we get a cytoplasm complete division where two separate daughter cells are produced. So let's look, this is a great slide to review. Here's interphase. So normal cell growth and development occurs during interphase, where you'll have the nucleus. The chromatin is loosely coiled DNA found in the nucleus. And the big thing in prophase is that in prophase, you'll notice that the nuclear envelope, kind of the boundary that would be surrounding this chromatin, is dissolved. So there's no more boundary around the nucleus. So the nuclear envelope is dissolving, and you'll start to see the formation of chromosomes during prophase as the DNA tightly coils to get ready to be separated. Metaphase is nice, and this is why I like metaphase, because you can see the chromosomes all lined up in the middle of the cell. That's metaphase. And then anaphase, the chromatids, the half parts of the chromosomes are being pulled apart. And you can see that here in anaphase. The chromosomes are being pulled apart to opposite ends of the pole. And you kind of start to see division, but not really. It's really in telophase where you'll start to see kind of a deep kind of indentation as two new daughter nuclei will form. And then finally, to complete mitosis, we have this, what we call cytokinesis stage, where a new kind of cellular membrane is formed between the two daughter cells. And that would be kind of the end of mitosis, when two complete new daughter cells are formed. Differentiation. So differentiation, a sperm and an oocyte in fertilization will unite to form a single cell, and then a great number of divisions that occur by mitosis will occur to give the trillions of cells of the body. And the process by which cells develop with specialized structures and functions is called differentiation. And during differentiation of a cell, some portions of the DNA will be active and others will be inactive. So we kind of have a sperm and an egg uniting to form one zygote. And then that will quickly divide into many, many trillions of types of cells. But the process that tells this cell to become a skin cell, this cell to become a kidney cell, that's called differentiation, when each cell will go on to form their specialized structures and functions. And we have lots of different cell types, which we are going to learn about in this course. We have bone cells, nerve cells, we have muscle cells, and we have red blood cells. So this process of differentiation, everything starts from one single stem cell, And then this one stem cell will differentiate into all of these other types of cells in the body. So apoptosis is just a part of life. Death is a part of life, and even cell death is a part of a cell's life. So apoptosis specifically is termed programmed cell death. It's a normal process by which cell numbers within various tissues will be adjusted and controlled. So for example, in a developing fetus in the womb, apoptosis will remove extra tissue between the fingers and toes so that you don't have webbed fingers or webbed toes. In some adult tissues, apoptosis will eliminate excess cells to maintain a constant number of cells within the tissue. Cellular aspects of aging, so what causes our cells to age and then die off? There's existence of what many believe to be a cellular clock, where there's just a kind of a limit on how long cells can live. There could be the presence of a death gene, we're finding out. There could be damage from DNA due to UV light rays or other chemicals. Free radicals could have been formed and not properly disposed of, or we could have damage to the mitochondria. Whenever there's any damage to mitochondria, and different toxins and poisons will target mitochondria, because if mitochondria are damaged, remember that the mitochondria are what create your body's ATP. Without ATP, your body will not survive. So any damage to mitochondria will kill off cells as well. A tumor, so just like we're talking about cells and cell death and cell growth, a tumor is an abnormal proliferation of cells where they're usually due to problems occurring in the cell cycle. So instead of the cell cycle kind of just maintaining constant interphase stage, the cell cycle is constantly dividing and it can't stop. Some tumors are benign, which is fine, and some are malignant, and those are the cancerous tumors. Malignant tumors can spread then through the body by a process called metastasis. So that takes us through the end of cell structure and function. And I'm just going to write a note here to remind myself. I'm just going to post in an announcement the cell cycle video that kind of gives you a visual representation of translation and protein synthesis and transcription. And that might help a little bit with those topics, because those are always a little tricky to take in. So, and we're just gonna, I'm gonna lecture for 10 or 15 more minutes. I just want to start the next chapter, which is on chapter 4. And just to get ahead, so we can be sure to finish this on Wednesday. So we talked about cells, and we're just going to start chapter 4, because chapter 4, as my Wednesday lab group knows, really explains tissues, or starts to explain tissues, which is what you're studying in lab these weeks. And tissues is any group of cells with similar structure and function, plus any extracellular substance surrounding them. So tissue is when cells come together to form a specific kind of component, but a tissue will involve the cells themselves, but then any extra stuff around the cells. And the word histology is specifically the study of tissues, and you can take histology classes where you just look in microscopes all the time. They're fun. So there's 4 tissue types in the human body, and they're shown here: epithelial, connective, muscle, and nervous. And we'll just try to get through maybe 1 or 2, maybe even just epithelial, if we can get through that in the next 10, 15 minutes, because again, the lab will kind of directly relate to this information as well. So epithelial tissue is any tissue that covers or lines a part of the body or an organ. Connective tissue is a very diverse tissue that makes up a part of every organ in the body, and I'll show you what I mean by that. Connective tissue is very diverse. Muscle tissue is a tissue that contracts or shortens, making movement possible. And nervous tissue is responsible for coordinating and controlling many of your body's activities, like your brain, your nerves, and your spinal cord. So epithelial tissue, also called epithelium, covers and protects surfaces inside and outside the body. And included in epithelial tissue are both exocrine and endocrine glands. And we'll talk about those 2 differences between those glands. But these are basic characteristics of epithelial tissue Epithelial tissue is mostly composed of cells. So what that means is in epithelial tissue, it's primarily just made up of cells and not a lot of extra stuff. And you'll see that in the other tissue types in the body, there's a lot of extra stuff. But epithelial tissue is composed mostly of cells. It covers the body's surfaces. It has distinct cell surfaces, where usually you have a cell surface that is exposed to a part and one that is not exposed. The cells and matrix are connected together very tightly. And this is an important one. Epithelial tissue is non-vascular, which means it does not have any blood supply to it. So no arteries and veins are going through epithelial tissue. And epithelial tissue is capable of regeneration because epithelial tissue lines and covers a lot of body surfaces. A lot of these cells will be constantly sloughed off, or there will be a lot of abrasion against these cells because they're lining surfaces where a lot of other substances are passing through. So here we have a look at epithelial tissue. This is a look at what we call a simple squamous, where you have a free surface that is exposed to kind of some sort of environment. And the cell surface, the bottom of the cell surface, is attached to the basement membrane. So this is a very thin type of epithelial tissue. Always underneath epithelial tissue, we'll have our connective tissue, which will have capillaries and blood vessels. But the epithelial tissue themselves do not have any blood vessels. Functions of epithelial tissue: because it's a covering tissue, it helps to protect underlying structures, act as a barrier, permits passage of substances, so only allowing certain things to pass through it. It secretes substances like your glands, and it also helps to absorb substances like epithelial tissue in your digestive tract, for example. We can classify epithelial tissue or name it based on the number of cell layers and then the shape of the most superficial cells, or the cells that are exposed. And the cell layers can be one single layer of cells called simple, multiple layers of cells called stratified, or kind of fake stratified layers, which we call pseudostratified. And then the cell shapes, you only have 3 options. Well, I guess 4 if we call our transitional shape, but they can be squamous, which is nice and flat. And this was a type of flat squamous cells. They can be cuboidal in shape, or columnar, like a column rectangle in shape, or they can have a special transitional shape, which can be kind of look like a balloon that can stretch and flatten in an area that needs to be stretched and then come back to its initial shape. So simple epithelium consists of a single layer of cells, with each cell extending from the basement membrane to the free surface. Stratified epithelium consists of more than 1 layer of cells, but only the basal layer attaches to the deepest layer of the basement membrane. And I'll show you examples of those too. And then pseudostratified columnar is a type of simple epithelium that appears to be fake or falsely stratified. So it really does consist of 1 single layer of cells where they're all attached to the basement membrane, but due to kind of the variations of the shape of the cells, the epithelia appears to be stratified or multiple layers. And then classification based on the shape of the cells that are exposed are most superficial. Squamous cells are flat or scale-like or fried egg-shaped. Cuboidal cells are cube-shaped, about as wide as they are tall, and columnar cells tend to be taller than they are wide, like a rectangle. Then I promise now we're going to go through names of each epithelial and then examples. And we'll go through several, and then once we get through epithelial, we'll take our break, because I promised you, I don't think our brains can handle much more than an hour of lecture, so we'll finish after epithelial tissue. So simple squamous epithelium, simple is single layer of cells, and squamous means they're thin and flat. So this type of epithelial tissue means that some substances can easily pass through it because it's very thin, but other substances cannot. So you'll find simple squamous epithelium in your lungs, which allow for gas exchange, oxygen and carbon dioxide to pass through. You'll also find simple squamous in kidneys, which filter waste from the blood, while keeping blood cells inside the blood vessels. So again, it's a type of epithelial tissue that allows things to pass through. And again, you can see a single layer of flat cells here. So this is called simple squamous epithelium. Underneath the microscope, you can see the simple squamous epithelium is lining this area shown here. So this is simple squamous epithelium. Simple cuboidal epithelium is a single layer, because it's simple, of cube-like cells that are really important for things like active transport, facilitated diffusion or secretion, and they have a much greater secretory capacity, so they can secrete things. So here we have a single layer of cube-shaped cells. You can see that here. So this is simple cuboidal, and this is a really nice picture. Simple cuboidal lines your kidney tubules. So you can see kind of this single layer of cube-shaped cells that I'm highlighting right here. The free kind of empty space in the middle is like the tube. So these cells will line a tube in the kidney to help secrete and absorb different things to create urine or absorb things back into the blood. So this is simple cuboidal epithelium. Simple columnar epithelium is a single layer, that's simple, of tall, thin cells. And the large size of these cells enables them to perform much more complex functions like secretion of different substances. Simple columnar epithelium, for example, in your small intestine, produces mucus and digestive enzymes to aid in digestion when food goes into your small intestine. So here we have a look at simple columnar. Again, you have one single layer of these nice column-shaped cells. You might see other, what we call goblet cells within them, secreting mucus, or other cells secreting other types of digestive enzymes, which will help aid digestion in your small intestines. So that's simple columnar, one layer of column-shaped cells. Pseudostratified columnar is kind of a fake stratified, but it's still one single layer. It will secrete mucus, which covers a free surface, and it will also often be covered with cilia to help move the mucus and anything that you accumulate in your throat or in your trachea towards your throat where you swallow it. So pseudostratified columnar lined with cilia will be important for kind of trapping anything harmful you might breathe. So you'll find this in your respiratory tract. Because then anything harmful that you breathe in will be trapped in this mucus being secreted by goblet cells. And then the cilia will kind of move that mucus so you can cough it out or you can swallow it. And you can see here why it's called pseudostratified, because the shape of the cells is not clear, but it's just a one kind of single layer of cells too. So this is found lining your nasal cavity, nasal sinuses, pharynx, trachea, bronchioles of the lungs. This is my one soapbox I'm going to get on for not smoking, because what smoking will do is it completely obliterates this type of epithelium to change, this nice goblet secreting mucus cells with cilia, which are good for trapping things that you might breathe in, whether that's pollution or anything. Smoking obliterates this just down to a simple squamous type of epithelium, so that smokers often develop a cough because they don't have the cilia and the mucus to help get their kind of junk out the way a non-smoker might. So what smokers will develop is a cough because they don't have the cilia and mucus to do that work for them. So they have to cough up anything harmful they might breathe in. I do know if you stop smoking, like right away, there's potential for this type of epithelium to grow back in your airways as well. And I'll get off my soapbox now. Okay, so then stratified layers. Stratified squamous forms a very thick epithelium because stratified means several layers. The deepest layers might look cuboidal because they're capable of dividing to produce new cells, but the naming is always based on the cells on the surface. So that's why we call this stratified squamous. And there's two types of stratified squamous. We can have keratinized, which are filled with keratin, which is a protein protective layer. This means they're dead at the kind of most superficial layer. Non-keratinized means they're not filled with keratin and they're still alive. And I'll show you the examples of that. So keratinized stratified squamous means the outer layer of the skin is comprised of keratinized squamous epithelium, meaning the keratin is a protein that fills up the cells when they die on the most superficial layer of the skin, and this reduces the loss of water from the body. So you'll find keratinized stratified squamous epithelium lining the surfaces of your skin. Non-keratinized stratified squamous, you can find it in the mouth, for example. It's moist because the cells are not filled with keratin, they're still alive, and it gives a kind of a protection against any abrasion, and it also acts as a mechanical barrier. Water can move across it a little more readily because it doesn't have that keratin in to kind of prevent water loss. So that's non-keratinized. So here's a look at the non-keratinized version, because you can see in the most kind of exposed surface or the most superficial surface, you have these, stratified squamous layers, multiple layers of flat cells. But in the most exposed or superficial surface, these cells still have a nucleus in them. So that means they're alive. So this is non-keratinized stratified squamous, because you can see these cells are alive. They still have a nucleus in them. A keratinized version would just kind of show a thick layer of keratin because all the cells would be dead. You wouldn't see individual nuclei in each of those cells as well. Stratified cuboidal is more than one layer of cuboid-shaped cells. It's a little bit rarer. We'll find it in sweat glands, ovarian follicular cells, salivary glands, and it helps to absorb, secrete, and protect. Stratified columnar is also rare. Stratified columnar is found in mammary gland ducts, the larynx, parts of the male urethra, to help with secretion, protection, and some absorption. And then transitional epithelium is kind of our last special type of epithelium that can be stretched, and the shape of the cells will change as this type of epithelium is being stretched, so it'll align parts of the body that stretch, like your bladder. It also helps to protect anything underneath the structure itself. So here's transitional epithelium when it's not stretched in an empty bladder, and here's when the tissue is stretched in a full bladder. So that's transitional epithelium. And let's see, I'm just going to see, I think we're going to stop there, and we'll kind of finish our discussion on epithelial tissue, talking about free cell surfaces, and then how these cells are connected together on Wednesday. So we'll finish this PowerPoint on Wednesday, and your first lecture exam on chapters 1 to 4 will be a week from today, next Monday. So I'll go ahead and stop the recording now.